Traditional breeding techniques of selection based on appearance or characteristics has been an incredibly important tool in animal improvement. If you compare the difference between a Chihuahua and a Great Dane, for example, you can see that breeders have made incredible progress based on phenotypes or the appearance of animals. When applied to our livestock population, techniques such as artificial insemination have greatly amplified the influence of genetically superior bulls. Since its adoption in the 1940s, milk production per cow has greatly increased, such that now we need only a little over 9 million dairy cows to produce all of the milk for US consumers, rather than back in 1944 when we had over 25 million dairy cows. If not for the genetic improvement that was enabled by artificial insemination, we wouldn't have been able to lower the carbon footprint of a glass of milk to a third of what it was in the 1940s. About half of the 369% increase in production efficiency in dairy cattle is attributable to the genetic improvement that resulted from AI. When it comes to genetic improvement of animals, there are many different breeding methods that can be used. These include relatively non-controversial ones like selective breeding, artificial insemination, and the use of DNA marker-assisted or genomic selection. But one breeding method, genetic engineering or genetic modification, has proved highly controversial, especially when it comes to producing food from such animals. As can be seen, there are many different reasons why animals are genetically modified, and there are different breeding methods that can be used to obtain that modification. There is even the opportunity to use genetic engineering to control insects that spread disease, such as the mosquito vector of dengue fever and the Zika virus. However, it is in the food production that the use of genetic engineering of food animals has the greatest potential. And the reason for that is that genetic engineering enables breeders to use useful genetic variation that does not exist in the species of interest. What I mean by that is that the techniques of recombinant DNA technology enable breeders to bring in DNA sequences that are not present in the species they are trying to improve. And this enables new traits to be brought into breeding programs. One trait of particular importance is disease resistance. Globally, it's estimated that approximately 20% of livestock production is lost to disease. If breeders, for example, can bring in a sequence of virus DNA that effectively vaccinates the animal and makes it immune to that virus, then this would have the impact of reducing animal disease, the suffering of the animal, use of antibiotics to treat the sick animals, and also prevent the production losses. I would argue that this is a really good use of genetic engineering. We are basically using genetics rather than chemicals to fight disease. These types of applications are where genetic engineering has a unique role to play, as it enables access to unique genetic variation to give animals traits that could never be achieved using conventional breeding programs. But combining genetic engineering with traditional breeding programs really could synergistically accelerate progress in sustainable breeding goals in a way that neither breeding method on its own would ever be able to achieve. Many animal biotechnology applications, including genetically engineered animals, align with sustainability goals, including improved animal well-being and welfare, efficiency, and reduced environmental footprint. And there really is a compelling need to have this type of technology used in animal breeding programs, given the projected increase in the demand for animal protein in the light of both human population growth and improved incomes in the developing world.